Hey everyone, and welcome. Today, we are diving deep into one of the most fundamental topics in biochemistry, amino acid metabolism. We're going to trace the entire journey of amino acids, from the protein in your food right down to their final destinations inside your cells. So let's jump right in. I mean, it's a great question, right? Especially when you're a first-year med student. You eat protein, it gets broken down into amino acids, they get absorbed, and then what? Well, they certainly don't just hang around. They enter this incredibly dynamic, super important system that's at the very heart of our body's entire economy. You know, this little poem from your textbook, it's actually the perfect cheat sheet for our entire discussion today. It lays out the two parallel journeys we're about to follow. First, the journey of that nitrogen-containing amino group, and second, the journey of the carbon skeleton that gets left behind. But before we can track any of those pathways, we first need to understand the starting point for everything. Let's talk about the body's central hub for amino acids. So get this. At any given moment, the adult body has a pool of free amino acids that adds up to only about 100 grams. Now, don't picture this as some quiet, static reservoir. It's more like a bustling train station with constant arrivals and departures. And here's a cool fact. About half of that entire pool is just two amino acids, glutamate and glutamine. And what's really mind-blowing is the scale of the turnover here. We might eat, say, 40 to 100 grams of protein a day, but our body is breaking down and rebuilding 300 to 400 grams of its own protein every single day. This really drives home a critical point. Unlike carbs, which we can store as glycogen, there is no storage form for amino acids. It's a use-it-or-lose-it system. Any excess has to be dealt with immediately. All right, so let's start with the first of our two big journeys. We're going to follow the fate of that amino group, the nitrogen part. And the body has to be incredibly careful with this because if that nitrogen builds up in the wrong form, it's seriously toxic. So the path for pretty much all amino acids starts with two main steps, one right after the other: transamination and then deamination. This whole process is designed to pull off the nitrogen and liberate it as ammonia, which is NH3. That ammonia is then sent off to get detoxified. Let's break down exactly how that works. First up is transamination. The best way to think about this is like shuffling. An amino group gets passed from one amino acid over to a keto acid. Now, this is useful for making new amino acids the body needs, but its main job, in terms of breakdown, is to collect all the amino groups from lots of different amino acids and funnel them into just one specific molecule, glutamate. So once all that nitrogen is neatly collected in glutamate, the next step is deamination. This is where the amino group is finally removed for good, setting free that highly toxic ammonia. And now, the body has a real problem on its hands, a buildup of poison. It needs a solution, and it needs one fast. And that solution is one of the most brilliant and elegant pathways in all of biochemistry. It happens almost exclusively in the liver, and it's called the urea cycle. Okay, let's hit pause for a second. It's a fair question, right? Why does the body bother with this complicated energy-guzzling cycle just to get rid of nitrogen? Why not just get rid of the ammonia directly? Well, the answer comes down to two things: safety and efficiency. The difference between these two is night and day. Ammonia is a potent neurotoxin. It's especially dangerous for the brain. Urea, on the other hand, is basically harmless. It's super soluble in water, which makes it perfect for transporting in the blood to the kidneys to be excreted. The body literally turns a dangerous poison into a safe, easily disposable waste product. The urea cycle itself is this amazing 5-step factory line that spans both the mitochondria and the cytoplasm of liver cells. It starts by taking that toxic ammonia and some carbon dioxide, runs them through a series of reactions, adds a second nitrogen, and then, pop, out comes a molecule of urea ready for disposal. But, this level of safety and detoxification isn't free. Each turn of the cycle costs the equivalent of 4 high-energy phosphate bonds from ATP. It's an expensive process, for sure, but it is absolutely essential to prevent the catastrophic effects of ammonia poisoning. Okay, so we've safely handled the nitrogen, it's been packaged up as urea and is on its way out. But what about the rest of the amino acid? That carbon skeleton that's left over is far from being just waste. In fact, it's a huge metabolic opportunity. Let's go back to our map. Now we're looking at the other side of the diagram.
After that amino group is gone, the leftover keto acid can be channeled into several incredibly useful metabolic pathways. So the key takeaway here is just how versatile these carbon skeletons are. They can be burned for immediate energy, for ATP. They can be used to build new glucose if your blood sugar is low. They can be converted into fat for long-term storage or made into ketone bodies. Or they can be used to create new non-essential amino acids. This is metabolic recycling at its absolute best. Now, for all of you future doctors, it is absolutely vital to know what happens when these beautifully controlled pathways break down. So let's connect all this biochemistry back to the clinic. If there's a genetic defect in any one of the 5 enzymes of the urea cycle, you get a metabolic traffic jam. Ammonia can't be converted to urea, so it backs up in the blood. This condition is called hyperammonemia. All of these disorders, like citrullinemia or hyperargininemia, are incredibly serious and can lead to severe brain damage or even a coma if they aren't diagnosed and managed properly. All right, I know that was a whirlwind tour through a pretty heavy topic, so let's lock in the most important high-yield points with a super quick recap and a couple of questions to test your memory. So if you remember anything, remember this. 1. Amino acid metabolism is a tale of 2 journeys, the nitrogen and the carbon. 2. The nitrogen journey is all about detoxification. Amino group becomes toxic ammonia, which becomes safe urea in the liver. And 3, the carbon journey is all about opportunity. That skeleton gets recycled for fuel, glucose, fat, or even new building blocks.
All right, first question. Quick recall. Which organ is the main site of the urea cycle? Give you a second.
That's right, it's the liver. The liver is the only organ that has the complete set of all 5 enzymes needed to get the job done. And for question number 2, once that amino group is gone, what are 2 things the body can do with the carbon skeleton? Well, you could say it's burned for energy, used to make glucose, turned into fat or ketone bodies, or used to synthesize new amino acids. Any 2 of those are perfect. And finally, to help you remember the whole sequence, here's a super simple mnemonic, TDUF. Transaminate, deaminate, form urea, and provide fuel. And there you have it. You now have a clear picture of how this whole system connects nutrition, energy, and excretion. It truly is the core of biochemical balance. Thanks so much for joining me.